We have another episode of the Beta Ellis series of lectures in the English language. Today we will be looking at the pioneer in bioresonance technology, which was Dr. Royal Raymond Rife. He is one of the geniuses of the 20th century, and he's certainly one of my heroes. Often when we deal with the technology in today's world, we often forget the people that have spent decades of their lives in laboratories working to make this technology available. So I think today I want to spend some time looking at who Dr. Rife was and what were his achievements. He was born in 1880 and he died in 1971. He was a pathologist, a scientist, an inventor, an accomplished musician, as well as an engineer. As a child, he had a very inquisitive mind. He was born May the 16th in 1880 to Zibe Raymond Rife Sr., who was a mechanical engineer from Nebraska, Ohio in America. His mother was Ida Mae Chain from Preston, Idaho, again from America. He was actually the second child, but his older sibling died in childhood. His mother also unfortunately died 8 months after giving birth to Rife. So given that his father was a very busy engineer at the time, his father asked his sister, Nina, to look after Rife, which she did as if he was her own. As she says in her own words, "I raised him and brought him up as though he was my own." So Rife lived with his aunt Nina from the age of 8 months old till 1905, when he was 17 years old. Thereafter, he went to Johns Hopkins Hospital to study medicine, and then he found that his main interest was in bacteriology, so he ended up qualifying as a pathologist. He did a lot of work in this field of pathology, and he spent a lot of time actually photographing many specimens for various institutions, such as the University of Heidelberg in Germany. And indeed, for this work, he was given an honorary doctorate in parasitology in 1914 by the university. Given his interest in microscopes, he also began to study optics. Indeed, he studied with the best, because from 1904 to 1908, he studied with senior Zeiss optics engineers in New York's Zeiss Works. Back in the 1850s, Zeiss and Abe, from the Abe the Abe condensers, the Abe microscope lighting, joined partners and became the leaders in microscopes and lens technology, and probably still are. Everybody is familiar with the Zeiss lenses, and so he did study with the best at the time. In 1912, when he was 24 years old, he decided to settle in California, and he married an Asian lady called Mamie Quinn of San Diego, and he dearly loved her. He wrote love poems for her, and she was a great support to him throughout his work. They never had children, and she died 45 years later in 1957. Because work was sparse at the time, Rife had to earn a living, so he ended up becoming a private chauffeur to Timken from Timken Bearings. He was a wealthy and philanthropic motor magnate, and as they were driving in the car together, Rife would often talk about his dreams of what he wanted to do in the field of science and bacteriology and parasitology. And Timken, who was an experienced business entrepreneur, realized that this man really knew what he was talking to and began to pay attention. Rife had a very, he had a seriousness and an integrity that did not catch Timken by surprise. He recognized the quality when he saw it, so he began listening. And what they decided to do is Timken, with another business magnate called Bridges, decided to set up a financial support by creating an endowment fund to finance Dr. Rife and his astounded research. And indeed, they set up in Timken's estate, they set up the Rife Laboratory in Point Loma in California, and Rife began working in his private laboratory. The laboratory you can see in the upper right-hand picture. Timken is in the bottom picture, and his wife is on the left. So from 1915 to 1920, Rife worked in his own private lab, identifying and classifying diseases, disease-causing microorganisms. However, he realized the limitations of the conventional microscopes that he was using at the time. So because microscopes could only magnify optically to around about 2,500 times diameter magnification, he set himself the task of improving upon this by building his own microscopes. And indeed, he built 5 microscopes, of which here we can see the number 3 microscope, which is a universal microscope. This was the most superior that he built, and this was all hand engineered in the laboratory. Every part of the 5,800 or so parts on this microscope was actually engineered by him and other engineers. It was a marvelous feat of engineering which defined optical laws and physics at the time, because it had a magnification that went way beyond 2,500 times, up to 30,000 times, with a resolution of around about 100 nanometers, which was unheard of at the time. So we are actually fortunate to have some real video footage of the Rife Research Laboratory. This is actually narrated by John Crane, who was Dr. Rife's engineer. And I would just like to spend a few minutes looking at this fascinating video back in the 30s, where we can get a real glimpse of the internal workings of his laboratory and just how sophisticated his research was back then. A lot of people think that, you know, Rife didn't exist or he didn't do the research, but I think this video footage tells it all. So bear with me, and we'll start the video, and then I'll come back after a few minutes.
Turn the light on. Oh, yeah, you got it. This is the Rife Research Laboratory. in Point Loma. This is Royal R. Rife, the research scientist of this laboratory. Here are views of the Rife Research Laboratory at Point Loma, California. This is narrated by John Crane. The principal scientist in this laboratory was Royal R. Rife, R-I-F-E, who had worked in research on cancer and many other diseases from 1920 to 1942. This is the chemical apparatus of the laboratory, part of it. The cultures are in this cabinet, which had to be protected from the radiation of the electromagnetic energy developed by Rife's ray tube instruments. There were many sterilizers in the laboratory, one being 15 feet long for the sterilization of glassware and other research items. These are hot water steam heaters. There was a complete machine shop in the laboratory for tool work, making microscopes, and providing any of the facilities needed for the research. This research was sponsored by the Rife Virus Microscope Institute, by Life Lab Incorporated, and by the John F. Crane Corporation, set up for continuing Rife's research. Every room was completed in itself. Nothing was taken from one room to another. Rife was a world traveler and a great marksman. He had 40 of the most beautiful guns made in those days. This is Rife's office, his desk. This is a vault where he kept some radium and other valuable materials used in the experiments on cancer. Rife had about 1,000 albino rats in the basement, which he used to grow tumors from a virus he isolated, a cancer virus, which was isolated and injected into the rats. These are steam sterilizers in a sterilization room. This is a surgery room in which the glassware and other surgery items were used in the operations on the rats by Rife. This is the main surgery table where the rats were, or the other animals were operated on. These are pictures of the surgical instruments used by Rife. Rife was assisted by many MDs that worked with him during this research. This is the optical room in which the microscopes and the elite's optical bench is shown in the background with his universal microscope, the Rife microscope published by the Smithsonian Institution and the Franklin Institute. This was one of 5 instruments that Rife designed and built with the help of his colleagues. This is a 1,000,000-volt X-ray. This is a stop-motion photomicrograph machine that was used to make delayed versions of the growing of flowers or the growing of viruses, the growing of plants, the growing of worms, which were all recorded. This is a Scania research camera, 35 millimeter, that Rife used in his work. And this is a number 2 microscope. A virus microscope was a prismatic instrument. And this is the number 3 universal microscope, prismatic type. That embodied all fields of microscopy, including dark field, polarized light, infrared light, ultraviolet light, and monochromatic beam light. The indicator dial moved around 7 times to move the object up and down 1 micron. And it was a very sensitive instrument. The ocular that you see on the left side in the middle of the instrument, when that was plugged in, made it a standard microscope. Now, in isolating cancer virus in 1931, Rife took an ulcerated human breast mass from the Paradise Valley Sanitarium donated by Dr. R.T. Hamer, who was assistant superintendent of the Paradise Valley Sanitarium and Hospital. Portions of this tumor were cut out and filtered through a triple-aught porcelain Berkfeld filter, which gave about 10 micron filtration. The virus could readily pass through a filter of this degree of closeness. The test tubes that are shown here are irradiated for 24 hours with neon argon or krypton gas, in which, like a fluorescent light, the radiation caused the virus to become virulent. Rife could then grow tumors in weeks instead of months in the art without this radiation. The test tubes were placed in a water.
Okay, that was one section of the video regarding the Rife laboratory. There are other sections that can be seen on YouTube. If you just go to YouTube and put a Rife laboratory, you'll be able to see all the sections, and you'll be able to see the rest of it, and it is definitely worth doing. Now, Rife was probably the first man in the world to be able to see living viruses under his viral microscopes, because of the resolution and the magnification that these microscopes were capable of. And not only see viruses, but also see living viruses, because under electron microscopes that have even greater magnification, yes, you can see the virus, but because it's bombarded with electrons, it kills the virus. So therefore you cannot see it in its living form. He was probably also the first man in the world to observe the phenomena of pleomorphism, where one bacteria will change to another bacteria or a fungus or eventually a virus. And he traced these cyclogenies with various microorganisms, And this is how he eventually discovered the BX and the BY virus. The BX was for the adenocarcinomas, the Y virus was for the sarcomas. And he was probably the first man in the world to use radio frequencies to eradicate not only the cancer viruses and cure cancer, but also eradicate many other microorganisms. He worked on approximately 55 different microorganisms during his lifetime and found the mortal oscillatory rates for all of these, and used his equipment to eradicate all of these. Around about the 1920s, he began working with electromagnetic fields to destroy the pathogens. He met Dr. Lee De Forest, who was the inventor of vacuum tubes, and De Forest was very helpful in helping Rife build his initial generators, as well as the tubes that were filled with helium gas to a pressure of 18 millimeters of mercury initially, and then he used argon gas and mixtures of gases. And he began observing that low frequencies could actually kill various microorganisms under the microscope. And this is where the amazing accomplishment of actually finding the resonant frequencies of these microscopes came from the invention of the universal microscope, which he used mostly. This surpassed the theoretical limits of optical microscopy in the 30s, and it gave unheard of resolutions of 17,000 diameters and more. He could also photograph and large, because of the fantastic resolution he was getting. He was able to electronically enlarge and provide magnifications of up to 300,000 electronically. So these ultra microscopes really began to trigger war on viruses, and there was no need to stain or kill the viruses or the other pathogens that were being observed, because these were all observed in their living state. So it was easy to set up the frequency instruments to actually observe the pathogens lysing and bursting when the mortal oscillatory rate was found. The mortal oscillatory rate is the frequency that resonates with the resonant frequency of the pathogens. So when you have the same resonant frequency and you have resonance, then the pathogens will absorb this energy, and they will lyse, they will burst. And this was observable in these microscopes, in these ultra microscopes, and this is how he identified the mortal oscillatory rates for more than 55 different microorganisms. Unfortunately, a lot of these went with him to the grave, because the medical mafia moved in after they heard that he was able to eradicate disease using electricity. This was definitely against the interests of the pharmaceutical companies, and so therefore they moved in, vandalized his laboratory, took many of his notes, poured him through the courts, and basically stopped his work, and also prevented other medical practitioners in America from having access to his frequency generators and his microscopes. In the 30s, after doing many experiments with the microscopes and identifying different microbes, including the BX and the BY virus, he actually took 20,000 tissue samples, which he cultured, he incubated, and he then took the viruses from these cancerous tissues, and he would then inject it into rats, and the rats would develop the tumor. He would excise the tumor surgically, and then he would use his bioresonance devices to cure the cancer in the rats. He did this 411 times. He was a bit of a perfectionist, and this was all monitored by some of the most eminent pathologists and bacteriologists of the time. Dr. Kendall was one of the probably world's leading bacteriologists of the time, who worked closely with Dr. Rife, and they successfully demonstrated the isolation of the BX virus to more than 50 research pathologists associated with the most noteworthy institutions. So this is real, folks. This was not, a dream, and the resonant devices that we have available today all came from Dr. Rife's work. And so we need to honor this man. We need to bow to this man who spent years and years and could have spent even more years researching if the medical mafia did not come in for their self-interest to destroy this work so that they can push chemotherapy and other toxic chemicals that clearly are not curing cancer today. Otherwise, we would have no cancer. And if money was placed into the research that Rife was doing, perhaps that would be the case today. Perhaps we would have no cancer. So he began working with electronic engineers. This engineer that we see here is Philip Holland, and this was back in 1938. This is a photo of the Rife Ray number 5, or the Beam Ray clinical machine, as it was called. This was the machine that was actually built by the Beam Ray Corporation, which they set up. And it was a commercial instrument. It was sold to medical doctors. There was a portable unit that came out of this, but there was also one that stood about 5 feet tall, had wheels, so you could actually move it around. So far, to date, a total of 3 of these original machines have been found and have now been reversed engineered. So we know exactly what frequencies they were producing, and these can now be reproduced in using amplitude modulation technology. We also have Rife speaking in his own words. There were Rife, when he used to communicate with other researchers, he would usually make tapes that he would then send to other researchers. And these tapes were found by Dr. Stafford in March 2000. They were sent to him back in 1957 from John Crane and the Rife Labs to help him with his research. And he had forgotten about them, placed them in a cupboard, and then they were found. And then the Kinnaman Foundation took on the task of electronically cleaning and improving the sound quality. And now we have 20 such CDs that anyone can purchase. And I strongly encourage people to purchase them. I think the last time I looked at the price was something around about $65, including excluding the postage. And this is the website, rife.org, rifecdorder.html, where you can go ahead and order these 20 CDs. And actually here Dr. Rife speaking in his original voice. I actually have a sample here, so let's spend just a few minutes listening to Rife's real voice. Here we go.
As far back as 1920, I conceived the idea and the possibility of when the causative agent of malignancy, so-called cancer, would be discovered and found and proven that it would be caused by a microorganism. Of course, reception that I received that far back from the medical profession and scientists was nil, but I kept at the work and I succeeded in eventually isolating a virus. First, I began sectioning tissues of every known type of malignancy. I sectioned over 20,000 of those, cut them down with a special microtome to very thin. Some of them were only a micron in thickness. I studied those under the microscope, and I eventually built my first high-powered microscope for the purpose of analyzing and checking those sections. The only result that I obtained over all those years was I succeeded in developing a very excellent technique of tissue preparation, but I never found an organism that I could say was the causative agent of malignancy. I went through with this, and then I developed other instruments of greater amplitude of magnification than the old original number one, which was very lacking in resolution beyond about 8,000 or 9,000 times. It was a lens microscope, and all of the air was extruded from the body and replaced with glycerin. The lenses were homogeneous with glycerin all the way through the whole thing, and the result was that in allowing the rays to separate and not cross in the interference band of reflection, such as they do in the ordinary standard tube of the regular research microscope, we held them apart, we separated them, and then brought them back together and picked them up again at a needle point. But, as I say, the resolution of this instrument beyond about anywhere from 9 to 10,000 times dropped off decidedly rapidly. We could go up on some occasions, on some preparations, up to 17,000 times, but we didn't consider the resolution anything out of the ordinary beyond about 8 or 9 or 10,000 times, depending entirely upon the specimen we were examining. With the results that those tissues gained us absolutely nothing. And as I say, I built other microscopes beyond this one. But that instrument we used up until 1931. With that instrument, Arthur Kendall and I, working jointly in the Pasadena General Hospital, succeeded in isolating what we classify the first filterable form of bacteria ever seen. It was isolated from the Bacillus typhosus from cultures that Dr. Kendall brought from his laboratory in Northwestern University in Chicago. And we succeeded definitely in isolating a filterable form of that bacteria. Now these filterable forms are very minute in size. The smallest of all is the one from the BX, which we isolate from cancer. We have never made a positive claim that that is the causative agent of cancer. But that is the smallest, and it's less than a 1/20th of a micron in dimension and highly motile. We succeeded in isolating this organism, this BX, using a Kendall media, known as Kendall media. It's made of a desiccated pig intestine, dehydrated down where it requires about 10 gallons of the original raw gut. when it's desiccated and run through the extractor and dehydrated to make about 1 pound of the original material. Now that is placed in a tube. We use about 4 grams, and then we use 10 to 20 cc's of a tyrode solution. It's a chemical series of salts. Now that is what Kendall used for the isolation or bringing out this Bacillus typhosus in the filterable state, which is published in the Californian Western Medicine in 1931, our joint report on that. Now these organisms, again, before we go farther, they cannot be stained with the aniline or acid dye stains that's used in the ordinary technique and method of staining tissue sections or even bacteria. Now those organisms are so minute and so delicate that they have to be stained with a frequency of light. Now after we built our first high-powered microscope, we had to build an illuminant unit to be able to see these different types of organisms and different types of tissue also. So we had to build this particular unit. We built a unit which I have a patent on, special lamp that will produce 2,000 candle power on a needle point of beam where it comes through the condenser. But between this source of illumination and the so-called substage condenser, we have a pair. On the universal microscope we have 4, but we use a pair of rotating wedge-shaped quartz prism. They're circular and they're wedge-shaped. And by rotating those, we bend the beam of light, that's what we termed the neutral or core beam of the illuminant unit, at different angles of incidence, so that it strikes the object under examination at different angles of refraction. There we see these particles at a different refractive index, and every one of them is entirely different. For instance, we find that the Bacillus typhosus, in its filterable state, is a beautiful turquoise blue body. We see them swimming through the field. They're highly motile. And incidentally, if the organism, the bacteria, is a motile bacteria, in most instances, the virus will be motile or highly motile, and vice versa. So we see these beautiful turquoise blue bodies swimming through the field. With that,
Okay, these are extremely interesting CDs. And there are many details of the research that Rife talks about on these CDs, because he's actually talking to other researchers. So go ahead and purchase a set for a little money, both to help the Kinnaman Foundation as well as to learn more about Dr. Rife and his research. He worked with many eminent doctors of the time. We mentioned Dr. Arthur Kendall, who was actually referred to as the American Pasteur. He was well renowned. He's on the left of the photograph. And then there were many others, Rosenow, Oscar Gruner, Ernest Walker, Arthur Yale, Dr. Couch, Dr. Mayer. These were eminent doctors of the time that all took tremendous interest in Rife's research and helped supervise the research, advise the research, etc. So Rife wasn't just someone who decided to do this work by himself and just publish the results. There were whole groups of eminent doctors that were supervising, and indeed when he eventually got to the point in 1939 when he had cured 16 metastatic cancers successfully. within 120 days, as well as many other patients with tuberculosis and other diseases that were epidemic at the time. He was invited to address the Royal Society of Medicine, which had recently corroborated his findings. He was requested to bring all possible film, slides, apparatus, which he did with great enthusiasm. And in the Journal of the Franklin Institute, volume 237, number 2, in February 1944, his therapy for cancer was actually published. So all these scientists, back in 1931, here they are at Dr. Milbank's house. Dr. Milbank was also a multimillionaire, and they all gathered in Dr. Milbank's house. Here you can see Dr. Rife, Dr. Kendall, Dr. Jensen, Dr. Heidegger, Dr. Ford, and 28 other doctors. And they all celebrated an end to all disease. However, unfortunately, the powers that be at the time did not like this idea of curing diseases using electricity. This was definitely against their self-interests. And so they sent in the medical mafia, and we know exactly the names of the people that were involved in destroying Rife. And this is indeed what happened. He was dragged through the courts. He was accused of practicing medicine without being a qualified oncologist. All the people that we saw in the photograph here just disappeared. It's as if they did not know Dr. Rife, even though many of them had actually worked by his side for many years. The American Medical Association, along with the equivalent FDA at the time, had decided to retract all the equipment, all the frequency generators that were sold to other medical doctors, and they were threatened that their licenses would be taken away if they did not return the devices. His laboratories were vandalized, and it's unfortunate that a lot of his research had disappeared. So how has Dr. Rife's research helped us in the battle against modern day diseases today? Well, many of his frequencies and their harmonics are now used in modern day Rife devices that use electromagnetic fields to eradicate pathogens, as well as helping to upregulate organ systems. So many of the devices that are sold by various companies, such as Dr. Ellis, actually use the frequencies that Rife had developed, or sub-frequencies, which are known as harmonics, which also have the same killing effect on the pathogens, but maybe take a little bit longer than the laser beam frequencies that Rife was discovering. Of course, the electronics are also different now. This is solid state. He was using amplitude modulation with old valves. The waveforms was somewhat different, but at the end of the day, most of these devices are using the Rife frequencies in one form or another. Here is an example of the Vita app made by the Russian company Dr. Ellis. Here, this is a device that is specifically designed for zapping parasites, bacteria, fungi, viruses, and other pathogens. It runs between 0.1 and 100 sorry, this one runs between 0.1 and 86,000 hertz, nearly 100,000 hertz. It has a USB stick that you can program these devices with. There are about 1,500 available programs for the DeVita app. When you purchase the device, it comes with 30 pre-installed programs. This is the 7th generation device. They have been on the market for some time. They work with 2 AA batteries, and these are some of the programs that you will get pre-installed when you buy the device off the shelf. There are also programs for detoxification. There are programs for helminths, worms, nematodes, tapeworms, ascaris, roundworms. There's also programs for a variety of different bacteria, such as Helicobacter pylori, different protozoa, such as Giardia, Lamblia, Trichomonas, fungi, such as Candida, Mucorrhizmosis, Aspergillus, and more. There's also a whole list of different viruses, such as the herpes family of viruses, which is the herpes Epstein-Barr, cytomegalovirus, Coxsackie virus. These are all the herpes family of viruses. There's anti-inflammatory programs, and so on and so forth. The other device that Deta Ellis produce, which is very popular, is the DeVita Ritme. The DeVita Ritme is really for balancing organ systems. It produces a different type of electromagnetic wave, a sine wave, that has the effect of upregulating the transmembrane potential or the voltage of different cells of different organs. So it really balances the organ systems. Again, on the same USB stick, you get another 1,500 possible programs for programming the DeVita Ritme with all the physiological systems and the organ systems. There's 30, again, programs pre-installed. The research and Deta Ellis have done approximately 117 clinical trials in various large clinics and hospitals in Russia. And they've worked on these diseases and these disease processes, the endocrine disorders, on high blood pressure and heart problems, angina, tachycardia, on kidney problems, kidney colics, etc., on gastrointestinal problems, on respiratory problems, bronchitis, asthma, pneumonia, headaches of all types. There are, I think, more than 30 different programs for different types of headaches with different underlying causes, neurological disorders, gynecological problems, ENT problems, tonsils, laryngitis, and so on and so forth. They also have developed bioresonance diagnostic devices, such as the Deta Professional here. This uses VRT testing, vegetative reflex testing, that Dr. Schimmel discovered based on Dr. Voll's work. So with these diagnostic devices, which are strictly practitioner devices, and they can only be used by practitioners who also understand the theory and also go through the clinical practice that is required to be able to operate these devices. This device not only is diagnostic, so it can identify all the pathogens in the body through resonant diagnosis, so it can identify the bacteria, the fungi, the viruses, etc. But it can also check the organ systems, identify organs that are weak or malfunctioning. It could also identify what organ has what underlying pathogen that may be causing the imbalance in the organ. It can trace the pathogenesis of disease, so the causal chain, by cross-correlating underlying causative factors, and so on and so forth. So it's a very holistic device. It also is a therapy device in itself, and it has 5 modes of treatment. So it has an electromagnetic, electroacupuncture, quantum light using colored light, different frequencies, again, light is frequency, and bioresonance, or a combination of all of these put together. So it is a powerful healing device as well. Bioresonance is beautiful because it actually takes the pathological oscillatory frequencies of the patient, even without understanding the underlying causative factors, inverts those frequencies and therefore cancels out the pathological oscillatory frequencies, and then it gives it back to the patient in a clean form. So energetically, it's cleaning up the pathological oscillatory frequencies. These are used by many thousands of practitioners. A lot of them are medical doctors around the world. In Germany, of course, it's very popular because Dr. Voll and Dr. Schimmel were both Germans. So they worked in that field and trained many other practitioners. But these are also spreading to many other parts of the globe. And we have Dr. Ellis to thank for producing this device, which is also very cost effective. Many of the other devices on the market that do similar things could range in the 30,000 euro mark. With this one, it's less than quarter of the price. So today I just wanted to touch upon the man that brought us this field that is now known as bioresonance or energy medicine, Dr. Royal Raymond Rife. He was definitely one of the pioneers. He was on the forefront. He started with nothing, and it looks like the cosmic forces had helped him to really come to a point where one is absolutely in awe of his achievements. Not only did he defy the law of optics and physics by developing the viral microscopes that had extremely high magnifications and high resolutions for technology of the day, so it gave him the opportunity of actually seeing living microorganisms without killing them with stains or bombarding them with electrons as you would do in an electron microscope. So by observing the living microorganisms and developing the bioresonance devices as well, the ray beam devices, and finding the specific frequencies by observing these microorganisms actually bursting in them under the microscope, when he found what he termed the mortal oscillatory rate, or the frequency at which these pathogens were resonating at. And this can be reproduced today. There are microscopes that are capable of similar magnifications and resolutions that are commercially available. Of course, they do cost hundreds of thousands to purchase, but assuming that the funding is there, these microscopes are available can be bought, and the frequency generators now have been perfected. As you probably heard me say previously, 3 of the original Rife generators have been found, and they have been reversed engineered by engineers, and they have also tracked down the equipment that Rife was using in those days, the Kennedy equipment from the California Electronic Museum, and these have been tested with spectrum analyzers to discover exactly the frequencies that were being emanated by these devices. So we have the electronics, we need the microscope at a cost, and if there's anyone willing to put some pounds, shillings and pence on the table, what we're saying is that this research is reproducible now to identify the specific laser beam frequencies, the multi-oscillatory rates that Rife unfortunately took many with him to the grave, for a number of incurable infectious diseases such as HPV, even herpes, even the common cold, even Ebola, the HIV virus, the hepatitis, and many more. Once these mortal oscillatory rates are